Hi class, we're going to focus on how we regulate our filtration rates in this recording. And we're going to talk about several different methods. It's going to be a little bit longer of a recording as we're diving into this material. If our glomerular filtration rate's too high, that means that we're producing too much filtrate and we can't reabsorb all the water in time. And that means that we produce more urine, the urine output rises, and we may become dehydrated or lose electrolytes from our body because we're urinating out too much water. If the glomerular filtration rate is too high, you have very clear urine. And it's not that, and this could be deceptive, because I remember as a college student, I started drinking lots of coffee when I was a freshman in college. And initially, my urine was very white after the first cup or two of coffee. And I thought to myself, woohoo, I'm super hydrated. But the coffee was actually dehydrating me by artificially inflating my glomerular filtration rate. So even though my urine color made it look like I was hydrated, I was actually becoming dehydrated because I was urinating out too much water. Now, if our glomerular filtration rate is too low, we end up reabsorbing waste in our filtrations of our filtrate, and that we have a tough time getting that waste out of our bloodstream, and we might have azotemia, too many nitrogenous wastes build up in our bloodstream. So if you ever are in a situation where you have kind of this brownish yellow urine, that's a really good sign that you need to go drink a couple glasses of water. Now, as we look at the glomerular filtration rate, we control it three different ways. We are going to have renal regulation, sympathetic control, and hormonal control. And with these three methods, we can ultimately control the glomerular blood pressure, the amount of pressure, hydrostatic pressure in the glomerular capillaries to control the amount of filtrate leaving the glomerular capillaries. Then we're going to talk about, in this recording, we'll talk about renal autoregulation, sympathetic and hormonal controls. So let's dive in and do autoregulation. Our autoregulation is the ability to self-regulate our nephrons. So each individual nephron can adjust their own rate of blood flow and their own glomerular filtration rates automatically without any external nervous system input or hormonal input. And this is going to allow for our kidneys overall to have a relatively stable glomerular filtration rate despite dramatic changes in our systemic blood pressure in the rest of our bodies. And we have two ways we can autoregulate. We have the myogenic feedback and the tubuloglomerular feedback. And in case you forgot, myo is a prefix meaning muscle. So this is a muscle-based feedback mechanism. And then we have the tubuloglomerular feedback mechanism, which we'll also discuss. As we look at the myogenic mechanism, it's based on the fact that smooth muscles have a tendency to contract when we stretch them out. So if we increase arterial blood pressure, that's going to cause the smooth muscle of the afferent arteriole to stretch out. And in response to that stretching, the afferent arteriole smooth muscle starts to constrict and reduce the blood flow back down to the homeostatic baseline, keeping our glomerular filtration rate more leveled out. And on the flip side, let's say we're in a low blood pressure environment, the amount of tension on the smooth muscle of the afferent arteriole is reduced. That causes the smooth muscle to relax and the afferent arteriole dilates, allowing for an increase of blood flow into the glomerulus. So whether we have high or low blood pressure, the smooth muscle of the afferent arteriole can contract and relax to help minimize the fluctuations in glomerular filtration rates. As we look at the tubuloglomerular filtration mechanism, this feedback mechanism is kind of a tongue twister right here. Tubulo, referring to the renal tubules, glomerular, referring to the glomerulus, feedback. So they interact with each other. So our glomerulus is going to receive a signal from the tubular fluid and adjust the filtration rate accordingly. So as the glomerulus is receiving this input, it can allow for it to regulate the composition of our filtrate, stabilize overall kidney output or performance, and allow for fluctuations in the blood pressure to be further leveled out and compensated for. And the tubuloglomerular feedback mechanism depends upon the juxtaglomerular apparatus. Juxta meaning that it's going to be overlapping two different things. It overlaps with the glomerulus. And it's a complete structure that we find at the end of our nephron loop, right where it has just re-entered the renal cortex next to the glomerulus. This loop, the nephron loop, right where it re-enters the cortex of the kidney, is going to come into physical contact with the afferent and efferent arterioles. And let's just skip ahead a slide here so you can see what we're referring to. I'm going to skip ahead 2 slides here. So here we have the nephron loop, and right when it gets back up into the cortex, it rubs up against the afferent and efferent arterioles. So this juxtaglomerular apparatus is right here. Let me back up a slide now. And within that juxtaglomerular apparatus, there are some structures we should highlight. We have a macula densa. This macula densa is some sensory cells within the nephron loop. And these sensory cells are going to have a cilia sticking out from them. And the cilia is going to be used to physically measure the glomerular filtration rates. If our glomerular filtration rate is too high, the macula densa is also able to measure sodium chloride concentrations. And as that we have that increase in sodium chloride concentration, that causes the macula densa cells to record or absorb more sodium chloride and secrete more ATP. That ATP is then going to be metabolized by some mesenchymal cells that are nearby, and turned into adenosine. And that adenosine is then going to be a transmitter that is going to stimulate the nearby glandular cells of the juxtaglomerular apparatus, or excuse me, those granular cells. The granular cells, also known as the juxtaglomerular cells, are a special kind of smooth muscle cell that wraps around arterioles. And those granular cells are going to respond to adenosine by constricting afferent arterioles. So to really just connect the dots here, when we have too much filtrate flowing through the nephron loop, the macula densa cells sense that and absorb sodium chloride and release adenosine, or absorb sodium chloride and release ATP. The ATP is absorbed by mesenchymal cells, which converts it into adenosine, and then adenosine causes the granular cells to have the afferent arterioles constrict. That reduces the amount of blood flow into our glomerulus and lowers the glomerular filtration rate. We also have mesenchymal cells that can constrict directly, causing the capillaries within the glomerular corpuscle to have a reduced amount of filtration. So let's recap on the figure here. Let's say we're producing too much filtrate. Too much filtrate is leaving through the proximal convoluted tubule over here. It's going to go away, whoop, doo, 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 and then eventually make its way back up into the nephron loop. And then the macula densa cells of the nephron loop right here next to the renal corpuscle sense the high glomerular filtration rate, absorb excessive amounts of sodium chloride, and then release ATP. That ATP is picked up by the mesenchymal cells, which are going to be nestled right here and extend into the renal corpuscle. The mesenchymal cells turn the ATP into adenosine, and the adenosine is going to be picked up by the granular cells. And those granular cells are then going to initiate a constriction of the smooth muscle of the afferent arteriole. Also notable, we have sympathetic nerve fibers. So if I activate the sympathetic nervous system, if I'm stressed out, we can also, through the granular cells, constrict the smooth muscle of the afferent arteriole to also reduce the glomerular filtration rates. Our granular cells also are going to store renin within them. And renin is a hormone that we can release to help raise our blood pressure. And the signal that causes us to release renin is when we have a drop in blood pressure. And that renin will then participate in the Renin-Angiotensin-Aldosterone system. We talked about that in a lot of detail in Bio 214, AMP1, and we'll talk about it in a little bit more detail as well, or we'll review it a little bit in just a little bit. As we look at this renal autoregulation, process that can help control the glomerular filtration rate. It's worth emphasizing that it's not going to be perfectly constant. Typically will fluctuate above and below a preset homeostatic balance or baseline. If our glomerular filtration rate falls, our macula cells are going to ultimately cause the afferent arterioles to relax because our mesenchymal cells are not going to be as activated. And that causes more blood to flow into the glomerulus, and that will increase the blood pressure within the glomerular capillaries and cause glomerular filtration rates to go back up again. So as we look at this, just to really summarize it, if we have too much filtrate being produced, we have excessive amounts of sodium chloride within our nephron loop. The macula densa cells secrete ATP in response to that. Mesengyals turn the ATP into adenosine. Adenosine stimulates the granular cells. Granular cells will then cause the afferent arteriole to constrict, and we make less filtrate in response. That is tubuloglomerular feedback. There's a lot of steps involved. It can be kind of intimidating at first. I know it was for me when I was a student, but I promise if you break it down into pieces, you can do this. It might be helpful to print off this slide and then chop out these little individual pieces and then just play kind of an order game where you drop them on the floor, drop them on your tabletop, and put them in the correct order. That may be one way that would help you to learn the material better. Let's talk sympathetic control. As we look at sympathetic control, we're going to have nerve fibers from the sympathetic nervous system go to the renal blood vessels. As the renal blood vessels are innervated, we find that epinephrine from the adrenal glands directly above the kidneys, directly superior to the kidneys, and activation of the sympathetic nervous system will both cause afferent arterioles within our nephron to constrict. We also constrict our afferent arterioles if we have lots of exercise or circulatory shock. Restricting afferent arterioles causes less blood to go into the glomerulus, and that will ultimately reduce glomerular filtration rates, and if we make less filtrate, we make less urine. So instead of sending blood to the kidneys to be filtered, ultimately it redirects the blood to the heart, brain, and skeletal muscles, so we are ready for our fight or flight response. And in these situations, highly stressful situations, our glomerular filtration rate can be reduced down to just a few milliliters per minute, as opposed to 125 milliliters per minute. We also have a lot of hormones that can help to influence our glomerular filtration rate. And this is going to primarily involve the Renin-Angiotensin-Aldosterone system. There is also antidiuretic hormone, which we talked about in ANP1, but we're going to focus a lot on this more complicated one right now. So we have the Renin-Angiotensin-Aldosterone mechanism. There are 1, 2, 3 key hormones that are linked to each other to help us control blood pressure and glomerular filtration rates. If blood pressure is dropping, we have blood pressure receptors or baroreceptors in the carotid artery and the aortic arch that are going to activate the sympathetic nervous system. Our sympathetic fibers are then going to cause renin to be released from the granular cells of the kidney. That renin is going to be converted to angiotensin, which is in the blood, and as a blood protein, or excuse me, it's going to convert angiotensin into angiotensin 1. Note here with angiotensinogen, because it ends with that GEN, this is a almost activated form of a protein. So when working with angiotensinogen, it's one step away from being activated and turned into angiotensin 1. And then that angiotensin 1 is going to be converted into angiotensin 2. And that's done by angiotensin converting enzyme, or ACE. And we have lots of ACE in our kidneys and our lungs. So again, we have a lot of steps here. Let's look at it in a diagram, so there's the same information just in diagram form. So blood pressure is going down. Our kidneys sense that and release renin. The renin goes into the bloodstream and will take angiotensinogen from the liver and convert it into angiotensin. Note it goes from a big protein to a smaller protein. So there's been a chemical change. That renin will cleave angiotensinogen and turn it into angiotensin 1. Most angiotensin 1 is converted to angiotensin 2 in the lungs. The lungs contain most of our angiotensin converting enzymes. So that angiotensin flows to the lungs, gets converted primarily in the lungs to angiotensin 2, and then angiotensin 2 ultimately is going to cause blood pressure to go up. And it causes blood pressure to go up 3 unique ways. First, angiotensin 2 goes to the hypothalamus of the brain, and it makes us feel thirsty. If we're thirsty, we drink water and we have more fluid in the blood. So step number 1, angiotensin 2 increases the thirst response. Step number 1, it increases thirst response. Step number 2, it causes vasoconstriction. So the smooth muscle within the tunica media of our blood vessels constricts, reducing the amount of space for our blood to occupy, causing blood pressure to go back up. And then step number 3, the third method, is going to be within the kidney itself. Angiotensin 2 can goes to the adrenal cortex and will help to activate the release of aldosterone. Aldosterone then travels to the kidney and causes us to reabsorb sodium ions. As sodium ions are reabsorbed in the kidney, that increases the amount of sodium in the blood, and our blood will then reabsorb more water through osmosis, ultimately increasing blood volume and increasing blood pressure. So as we look at that angiotensin 2, it increases blood pressure very actively. It causes that vasoconstriction of blood vessels throughout the body, causing blood pressure to rise throughout the body. It's also going to constrict the efferent arteriole, that efferent arteriole that carries blood away from the glomerular capillaries. And if this efferent arteriole is constricted, if we pinch it off, that's going to make it so that we have to force more filtrate into the glomerular capsule, or glomerular corpuscle, so that we increase filtration rates. Our angiotensin 2 is also capable of lowering blood pressure within peritubular capillaries. And if we have lower blood pressure within peritubular capillaries, we absorb more sodium chloride and more water from the filtrate. In other words, angiotensin 2 helps us to make our filtrate more concentrated. Angiotensin 2 goes to the adrenal cortex, causing the cortex to release aldosterone. And just as I mentioned on the last slide, aldosterone makes us absorb more sodium in the distal convoluted tubule and collecting ducts. As we absorb more sodium, that is going to increase the rate of water reabsorption through osmosis within the proximal convoluted tubules. And then finally, angiotensin 2 stimulates the posterior pituitary gland to release antidiuretic hormone, ADH, also known as vasopressin. And ADH will cause us to reabsorb water directly through aquaporins within the collecting duct. So angiotensin 2 works many different ways to cause us to have our blood pressure to go back up and urinary output to go back down. It's kind of a lot. I recommend that you make a bunch of little post-it notes or flashcards for all these individual steps and just practice shuffling them and then putting them in order to help you memorize these sequential physiological sequences. And I need to go take a drink of water right now. All of a sudden, I'm pretty thirsty. If you have any questions or comments, please let me know. And as always, happy studies.